It's a pleasure to be here, and as John said, I'll be telling you about phasing and imputation algorithms. But before I start, I kind of want to get a sense of the level of familiarity all of you have with phasing and imputation, so I can kind of gauge speed accordingly. So just show of hands, who knows what phasing and imputation mean? Okay, almost everybody. Who has ever run a phasing or imputation algorithm? Anyone? About 4 or 5 people. And who has ever implemented a phasing or imputation method? 2. Okay, cool. Great. And then finally, who has heard of a hidden Markov model? Show of hands. Almost everybody. Okay, so you obviously didn't come here to learn what a hidden Markov model is. That's good. In that case, I won't bother going through the details of that. And then finally, who has coded a hidden Markov model? About half. Okay, great. So that's good, useful to know. Well, I'm going to start with some terminology first. And I guess I won't dwell on this because I think you just told me that you all already know what phasing and imputation mean. So I'm just going to start by sort of framing this mathematically so that we can kind of have a shared terminology as we go through this talk. And so for my purposes, I'm just going to consider phasing and imputing biallelic SNPs. A little bit bigger, okay. I will try to write a little bit bigger.
Or a lot bit bigger.
A lot bigger, okay. I'll try to write a lot bigger. Well, what do I mean by this? I mean, I'm just only going to be focused on variants in which you either have one of 2 alleles, a reference allele or an alternate allele. We're not going to worry today about multi-allelic variants. And we're also not going to worry about indels or other types of variation until Ronen gives the actual MIA seminar, and then we'll worry a lot about those things. For now, we're just talking about biallelic SNPs, which means that we can represent our data, our genetic data, as just, maybe I should add one more thing, diploid genetic data as just strings of 0s, 1s, and 2s. And we can also represent haplotypes as strings of 0s and 1s. Okay? And furthermore, I'm going to write out, I'm going to write a haplotype for an individual person as literally a string, 0, 1, 0, 0, et cetera. So this is a haplotype. And I'll write a diplotype, which is just an individual's sum of their 2 haplotypes as a string of 0s, 1s, and 2s. Oops, I have to make that consistent with my haplotype. Okay, so that's my mathematical terminology so far. And now, what are we doing when we're trying to phase? Phasing amounts to separating a diplotype into a sum of paternal plus maternal haplotypes. And imputation, I'll get into imputation later. I've written down a diplotype, I've written down a haplotype. What would phasing this diplotype amount to? It would just be saying, maybe this diplotype was formed as the sum of this paternal haplotype plus the corresponding maternal haplotype, which would just be what I would get from subtracting the 2. Great. So, that's the problem we're trying to solve in phasing. And I'm going to tell you about methods that we can use to do this computationally. But I think in order to really kind of grasp why the methods are useful and why they're doing a good job, it helps to just sort of pause for 30 seconds and think about how would you go about trying to phase a set of diplotypes. If I gave you, let's say, all of the genotype data in the 1,000 Genomes project, 1,000 individuals, unphased, in a section of chromosome, how would you go about trying to figure out how to divide these diplotypes into pairs of paternal and maternal haplotypes? And I guess a lot of you are somewhat familiar with algorithms, but just assume for now that suppose you'd never heard of a phasing algorithm before. What would you think? What might you try to do? The zeros and twos are easy. Yes, that is true.
I guess you would take like the correlation matrix between the SNPs, and if you saw two SNPs that were very correlated, you might expect them to be on the same haplotype.
Yeah. Actually, I'm just going to write down a list of these ideas because these are actually really good for intuition. So first thing was zeros and twos are easy. That's actually quite important. There's nothing to do. You know how to split them. Another idea is basically use SNP correlations, or in other words, LD, linkage disequilibrium. To estimate which pairs of consecutive alleles go together. Great. Yeah.
Look for pairs of individuals that are identical by descent.
Yeah. So look for pairs of individuals that share DNA identical by descent. Usually abbreviated IBD. And I want to kind of talk a little bit more about this and to flesh that out a little bit. So in the extreme case, if you had sequenced somebody's parents, well, then phasing the individual would not be all that hard. It's not completely trivial. So suppose that we have genotypes of parents. So let's say I have a paternal plus maternal, and I'm trying to phase this diplotype 0, 2. Well, if you told me that the paternal genotypes were, let's say, now I have to be slightly careful. If you told me the paternal diploid genotypes, then we could use observation 1, the zeros and twos are easy, to say, well, OK, at this third position, I know that the father had a 2, which means that the father's 2 alleles must have been 1 and 1, which means that the father passed a 1 down in the third position. which means that the mother passed the 0 down to the 3rd position. And so you can kind of just read off the phase inheritance whenever you have a homozygous allele for either the father or the mother. Now, to expand upon that, Ronen suggested something slightly fancier, which is that you could make do with not having parents, but if you just had some distant relatives, like somebody's, or not that distant relatives maybe, let's suppose that you had somebody's sibling, and that sibling had inherited the same tract of DNA from your father, then you could kind of use that sibling information to sort of stand in for a paternal reference, for a paternal haplotype. OK, so far so good? Cool. So this is sort of I think these 3 observations are actually probably the 3 most important observations in trying to go about performing computational statistical phasing on a large genotyped cohort. Namely, that the 0s and 2s are easy, so you get some sort of information for free. Second, that you want to use essentially linkage disequilibrium correlation information between nearby SNPs. That's going to give you some statistical information about how to sort of phase consecutive SNPs. And then thirdly, if you happen to be so lucky as to find long tracts of genome that are shared identical by descent between pairs of individuals in your data set, well, then you can further leverage that information in order to phase by sort of reading off haplotypes off a set of matching individuals. So now, I guess maybe I'm going to start moving into the actual HMM piece of this. And I guess I'll move over to this board. So you could basically think of hidden Markov model-based phasing as a fancy way to combine observations 2 and 3. So hidden Markov model-based phasing. It's just a fancy combination of observations 2 and 3. Because so far, well, we've just sort of put down some observations. We kind of want to use SNP correlations. We kind of want to use identical by descent sharing. But of course, in order to actually make this into an algorithm, a piece of software that we can run, we need to kind of codify this, and we need to sort of write down some sort of a model with which we can then use, a model that incorporates these pieces of intuition, but in a sort of statistically solid way. And so, well, there's basically one model that kind of underpins almost all of the work that has been done in the past 10, 15 years on phasing and imputation. That model is called the Lee and Stevens HMM. And the reference is Lee and Stevens, 2003 genetics, I think. Should have refreshed my memory before I came here. I think that's right. Well, what is the Lee and Stevens HMM? Again, it's basically a way to put this intuition into action. And the idea is to define a generative model for producing haplotypes, where I said by producing, I kind of mean equivalently sampling haplotypes, given a reference set of haplotypes. So let me try to make this a bit more explicit. You could imagine, I told you at the beginning, I might be giving you the 1,000 genomes data set and I asked you to phase it. Well, the 1,000 genomes data set, if you actually download it it's already phased. And so imagine now that somebody gave you the 1,000 genomes data set, which is already phased. And now I'm trying to define a generative model that tells you, what does the distribution of haplotypes look like, of new haplotypes that I might sample from, well, anybody in this room? What would that statistical distribution look like? And that generative model is then going to allow us to do phase inference, posterior inference. So, well, what's this generative model? Well, let me write down my reference haplotypes as, now I'm just going to write these reference haplotypes as horizontal lines. You can imagine that these are actually bit strings, just like the haplotypes that I wrote on the left board, but I'm not going to take the time to write down all the ones and zeros. And so now I want to, how do I sample a new haplotype? Which is just going to be an equal length bit string. And so the Lee and Stevens generative model basically just says, well, the only thing I know about how DNA looks is what these reference haplotypes look like. The most simple-minded thing I could do, if I really knew nothing else, would be to just say, I've got 2,000 reference haplotypes on my hand. I can sample them just by picking one of them randomly. That's my generative model for DNA. It actually wouldn't, it would be much better than if I just generated random strings of zeros and ones. It wouldn't be horrible. But, well, obviously that wouldn't be that representative of the people in this room. That would be representative of the haplotypes that are in 1,000 genomes. Fortunately, we can do much better than that in a very simple way, which is that, well, we kind of, well, so why was that not a completely unreasonable thing? It was not a completely unreasonable thing because people are, if you sort of follow a lineage tree up generations, tens, hundreds, thousands of generations, you'll eventually see that people share DNA with other people. And at any point at any position in my genome, if we go back far enough, we'll find somebody in 1,000 genomes who shares some maybe a couple kilobases of DNA by descent 1,000 or 3,000 years ago, or 1,000 generations ago, maybe. And so somehow there's information in the reference haplotypes that we want to use in modeling what DNA looks like, what haplotypes look like. But we can't just take them wholesale. We kind of know that the sharing only occurs locally in small sections of genome. And if we want to kind of probabilistically model what the universe of haplotypes looks like, they're going to kind of be a patchwork of chunks of DNA that's copied from different reference haplotypes. So what might such a patchwork look like? Well, maybe, actually I'm going to recolor this as blue. So I'm going to create a new blue haplotype. And maybe that blue haplotype kind of matches the second haplotype at the beginning, and it matches the third haplotype after that, and it matches the first haplotype after that, and it matches the last haplotype at the end of this chunk of chromosome. And again, well, what is this saying? It's basically saying that if we go back 1,000 generations or 10,000 generations, then this blue haplotype shared a common ancestor with that second reference haplotype in 1,000 genomes. So this is a much better model than just sampling randomly from the reference haplotypes. And the reason why it's a much better model, well, first, it incorporates this basically recombination. That's kind of what is causing the fact that you're sharing little chunks of DNA. And so, well, now the natural thing to do is just sort of imagine linking these guys up. And now this starts to look a lot like a hidden Markov model, where the hidden Markov model states correspond to reference haplotypes. Transitions correspond to recombination events. And then there's one last piece of this Lee and Stevens hidden Markov model that I haven't told you about, which is what are the emissions? And the emissions are just the observed allele. And so, well, I told you these reference haplotypes are in reality bit strings. And so they should kind of be labeled with zeros and ones along them. So I've written down 0, 1, 1, 0, 1, 1, 0, 0, 1, 1. Now, the Lee and Stevens model tells you that your blue haplotype that you actually sample should look a lot like 0, 1, 1, 0, 1, 1, 0, 0, 1, 1, but it allows you to have a possibility of changing the allele that is sitting on the reference haplotype. So maybe my actual blue, it's almost the same, 0, 1, 1, 0, but maybe then at this position, it actually mismatches the reference haplotype's allele. And why is that important? That's important because in reality, you could have all sorts of things happening that's causing the mismatch. You could have a mutation that occurred somewhere on the path to the most common ancestor. You could simply have genotyping error that you mismeasured either the reference allele or this target allele. And so really, I guess I want to kind of highlight that the two key pieces of this Lee and Stevens model are precisely the fact that it has these transitions corresponding to recombination events and emissions corresponding to observed alleles being possibly different from the reference, taking into account mutation and taking into account error. And these two aspects that I've starred are really the two most important things that you can model when you're modeling haplotypes. The transitions kind of make the model realistic because you have recombination events modeled, and the emissions make the model robust because you're now not so worried about having small errors. And I guess the beautiful thing about this hidden Markov model is that it incorporates both of these two sources of real or technical artifact variation, and it does it in a way that's computationally feasible. That's kind of the beauty of the hidden Markov model. So let's see, I guess I'm about half done with my time, and I'll now start talking a little bit about algorithms, what you can do with this hidden Markov model. But yes, any questions?
With the references, do they collapse down segments that are identical between two reference haplotypes, or do they use all the reference haplotypes as is?
Yeah, so the question is, let's imagine that we have a big reference panel, even, let's say, 1,000 genomes. And the question is, if you have haplotypes that are identical or locally identical, maybe in some kilobases or megabase range, then does the model kind of collapse together that information? And I guess you must be asking this from a computational perspective, because from a theoretical perspective, I guess it doesn't matter. And that's kind of precisely the point, that if you had sort of a collapsed state that kind of integrated together information from 10 different haplotypes that looked exactly the same, then it wouldn't matter for the probabilistic inference whether you modeled that as one state or 10 states, but it would matter for computational.
There are two possible states that are basically identical. We just pick randomly one of them?
Yeah, so if you had two states that were identical locally, then once the model kind of got there, it kind of wouldn't really care which state it was in, and those would probably get sort of they would contribute kind of an equal amount of probabilistic weight in whatever you were doing. But in terms of the computational considerations related to sort of can you or do you collapse haplotypes, well, the answer is basically yes, you do, in all sorts of different ways. And that's kind of become more and more important over the past maybe 2-3 years as these data sets have really expanded dramatically in size. And so the most recent version of both Beagle and Minimac two of the state-of-the-art imputation algorithms do some sort of a collapsing. The phasing method I developed, Eagle, it collapses in sort of a slightly different way. But collapsing is definitely one of the key pieces of this. Oh, yeah, thanks. Any other questions before I start diving into computation? Well, actually, maybe I should write that down explicitly. The input is a sequence, a diplotype sequence of 0s, 1s, and 2s, along with a reference panel of 0, 1 haplotypes. And the output is a pair of 0, 1 sequences that sum to the input diplotype. Great. Well, I just told you what the least evens hidden Markov model is. It's a generative model for producing haplotypes given a reference panel of haplotypes. That's sort of almost what I need. Now, the thing is I'm trying to perform inference given an observation of a diplotype sequence, not a haplotype sequence. But, well, fortunately, that's not too hard. I need to just sort of modify the least evens HMM slightly and make a diploid least evens HMM. And all that does is it just independently sample a pair of haplotypes and then add them together. And so now I've defined a perfectly good generative model that tells you what the population distribution of diplotypes might look like. Just sample a paternal haplotype by walking through this HMM once, and then walk through the HMM again and sample a maternal haplotype. And add them together, and that's what the universe of diplotypes looks like. So, OK, well, that's great. So now what this does is it has defined a population distribution on diplotypes. And so that means that this is basically a prior. I have, as my input, a diplotype sequence I'm trying to phase. That's my observation. And now the inference technique is just what you always do when you have a prior and an observation, which is just combine them together, and we can get a posterior. And well, what might we want to do with this posterior? We could find things like a maximum likelihood phasing, where by maximum likelihood phasing, I just mean the most likely pair of haplotypes generated by Lee and Stevens, or by most likely, I mean the sort of highest probability weight pair of, or highest probability way to decompose my input diplotype into a pair of phased haplotypes. Or we could do fancier things like some sort of a expectation. But maybe I won't go into the details of that. Let's for now just think about the possibility of trying to find maximum likelihood phasing. OK, well, when I asked at the beginning, half of you said that you have worked with HMMs before. So how do I do it? OK, you start by initializing a starting probability. Sounds reasonable.
Take that starting probability and times it by condition. So you do the first step and then iterate out the
Yeah, so I think what you're getting at, and maybe I need to be a bit more explicit about what we mean by starting. I think you mean left to right. You're sort of going to walk left to right across your HMM. And now I'm going to just kind of draw the canonical picture of what an HMM looks like. So here's kind of my most general picture of what an HMM looks like. Well, now this is the HMM. We have an observation. And we're trying to find a maximum likelihood path through the HMM that could have produced this observed sequence. And so, right, so how do we do this? Well, what you're starting to describe is presumably a dynamic programming-based way to perform posterior inference on this HMM. And maybe I should sort of preface this by dynamic programming approach to posterior inference on HMM. So initialize starting probability on the first state, on the leftmost state. And then we kind of walk left to right, accumulating the total probability of reaching a state. So raise your hand if this is sort of familiar to you. OK, I guess it's not really all that familiar. So maybe I should spend a couple minutes talking about what's going on here. Well, basically, the basic idea, or the problem, I guess the problem is we want to find most likely path through the HMM. And the difficulty is that, well, there are exponentially many such paths. And well, that's kind of obvious because as you walk left to right across your HMM, at any point you can have each of the possible transitions into each of the possible states. And so the number of possible paths just kind of explodes as you walk left to right. And if we're trying to find the maximum likelihood path, then in theory we would need to consider all of those. Maybe I should put in some variables on the numbers of states. Let's suppose that the dimension n is the number of states, and m is the number of SNPs. And so I guess what I'm saying is that there are exactly n to the power m such paths to consider, and we can't possibly consider all those. Yes?
You have to specify a emissions probability in terms of like an error rate and a transition probability in terms of the combination rate like ahead of time, or is that for?
Yeah, so the question is, let's see. I guess I didn't actually ever write down any variables that that define transition probabilities or emission probabilities. But the question is, do you have to declare those in advance, or can you kind of learn them as you go? And the answer is basically, if you have a very small data set, then you kind of need to learn them as you go because, well, you kind of need to extract as much information as you possibly can and make the model as precise as you possibly can. And so the early phasing algorithms from Matthew Stevens and his colleagues all did that kind of sort of iterative update of the model parameters as the algorithm progressed. In recent years, we're kind of at a situation in which the amount of data kind of trumps the prior because we're looking at trying to phase tens of thousands, hundreds of thousands of individuals at a time, and then you can be a little bit more fast and loose that you don't really have to worry about it. And so modern algorithms, you basically always just say, the error rate's about 0.1%. I'm just going to hard set that parameter and don't worry about that. OK, well, returning to performing inference on this HMM, I need to, again, walk through the HMM, or I want to find the best walk through the HMM, but I want to do it in a way that doesn't cause me to compute for the rest of eternity by looking at n to the m possible paths. And so, well, what can I do? This is where this dynamic programming trick comes in. And the trick is basically, I'm going to, actually, I guess I wrote one word wrong, which is kind of important. I'm going to walk left to right across my state space and accumulate the maximum probability of reaching each state. And so now, so what does that look like? If I start at my top leftmost position, then now, assuming that I start there, and let's suppose that I start off with an equal probability, 1/3, 1/3, 1/3, of starting in any of those states. So now what am I trying to do? Well, ultimately, I'm trying to find the best possible, maybe I should use one more color, the best possible path through this HMM that maximizes the sort of total probability weight that I've gained as I walk through this HMM. And so I can do this in a way that doesn't actually require me to consider all, well, or to literally walk through all possible exponentially many paths by just tabulating, by walking one position at a time, and at each point just tabulating what is the maximum probability that I could have obtained at each of these states. Yes? You thought the algorithm that I was relying on used EM instead of Viterbi.
Yeah, the maximum likelihood would be more like a Viterbi approach rather than a sum of the prior probabilities?
Yeah, so I realized as I was talking that I was sort of mushing together various things, which you have now called me out on appropriately. And I realized that with 15 minutes left, I probably should just stick with a maximum likelihood and sort of walk through this, given that it's not all that familiar to the people in this room. So I think I'm kind of going back on what I just told you, that there are a couple of different ways in which you could perform posterior inference. And for now, since the dynamic programming itself doesn't seem to be that familiar to everyone in this audience, I'm just going to describe sort of the most simple thing that you could do, which is to find this single maximum likelihood path through this exponentially large space of possible paths, which was also why I edited this word here. So I've now decided that I'm going to just look for the single most likely path through this exponentially large space. And so the way to do that is to just look to store at each position two things. One, the maximum probability obtained by any path reaching the position. and 2, which previous state, which actually I should be quite specific about position. Here I mean I want to store the maximum probability obtained by any path reaching the state. And here I want to say store at each position for all n states. Store the maximum probability obtained by any path reaching each of the n states. And then secondly, which previous state produced that maximum possibility, maximum probability. So what do I mean here? I started off with 1/3, 1/3, 1/3. Now I'm just going to consider all 9 possible ways that I could have transitioned from my first state to my second state. So there were, if I now look just at this top state in the second column, well, there are 3 ways that I could have gotten to this top state in the second column. And they each have some probability associated with them. And so now I'm just going to take the maximum over the 3 possible ways that I could have reached this second state. And let's suppose that the maximum, let's see, I'm going to use one of these other colors now. Let's suppose that the maximum was achieved by coming from the second state. Then that's the information I'm going to store. I'm going to store basically exactly what is illustrated in this picture, where it came from, and then how much was the probability that it got to. And so, well, I'm going to store that for state 1, state 2, state 3, and then proceed one more, do exactly the same thing, store what is the maximum probability that I could have gotten, the maximum probability of any path that could have reached this top state in column 3, where did it come from, and then continue walking left
So,
to right.
Peru?
Yeah.
So when you go to across that first red arrow, you now are at this state in the second position. There's also some emission probability.
There's also
you have to multiply that in
Yes.
as well before you then proceed to the next step, right?
Yes, that's exactly right.
Okay.
And so I'm kind of being fast and loose and not writing down any equations, but
Right.
that's exactly right. You have to incorporate both the transition probability and the emission probability. And that's really the only way that you're going to ever get any information, because otherwise then all of the paths would be equally likely, because you have the same chance of jumping to each of the states from each position.
Yeah, actually, can you share a little intuition about the structure of the transition matrix? So
The
there's some
structure of- yeah.
some probabilities stay the same, some you probably move somewhere else. Do we want that to be uniform? Do we want to learn that?
Yeah. So the simplest thing to do is to just have right, I guess I never wrote down what exactly that transition matrix looks like. So the transition probabilities are really just uniform. So we have a probability P of jumping to any other random state. And typically, we just make this uniform across the other random states, and then a probability 1 minus P of staying in the current state.
And can you take advantage of the structure of that transition matrix in the algorithm?
Yes, so that's actually probably the last thing I want to kind of say, which is not completely obvious from what I've said so far. So it's actually very important to use that structure of that transition matrix. And so this part, maybe for people for whom all of this is old hat, there's maybe something slightly non-trivial and new here. So far, well, first, I guess I made a point of the least evens HMM being haploid. It generates haplotypes. And then I said, well, when I'm phasing, I need to generate diplotypes. But no problem, I'm just going to sample twice from the least evens HMM. That gives me a generative model for diplotypes. Now, then I kind of threw all that under the rug when I went to sort of walk through slowly what an HMM inference method looks like. And just sort of sort of gave you a canonical example of a couple, like 3 different states. But now there's a trick here, which is that how many states does this diploid least even HMM actually have? Well, if I started with, let's see, where do I want to go? Maybe I'm going to go here. So the number of states in the Lee Stevens HMM, diploid HMM, is actually n squared, where n equals the number of reference haplotypes. And that, yes?
I have a question. So is there a way for the transition matrix to account for how long kind of the sequence of ones or zeros in is there because maybe having it too long of a sequence does not make sense. So I'm not seeing it, maybe I'm misunderstanding it, if there's a way of incorporating that then.
Yeah, so I guess the question is, would you want to sort of modify the transition probability with the knowledge that you have kind of copied from a single haplotype for a long time? Would that perhaps make you think, oh, maybe I've copied from this haplotype too long, and I should up the probability of jumping to a different haplotype? So the answer, well, I can tell you 2 things. One thing is that, well, you can't do that within an HMM framework, because that actually completely violates the assumption of the HMM being Markovian. That is, it's sort of, you can't ever use any information about what came before you. So computationally, you can't do that. The interesting thing statistically, or population genetically, is that actually, you actually almost want to do the opposite thing, that if you've copied for a long time from a haplotype, then you actually should decrease the weight of jumping, because that kind of means that, oh, well, if I've copied from this other haplotype for 50 megabases, then that's probably actually my dad, and I should just continue copying through for the rest of the chromosome. But we can't actually incorporate that in HMM. All right, so let's see. In 3 minutes, what else can I say about computation and Lee Stevens, and specifically whether you can take advantage of the structure of this transition matrix? Well, so if we started off with n reference haplotypes, then the number of states in my HMM is actually n squared, because I need to have a pair of states, one for the father and one for the mother. So if I have n squared states in my diploid HMM, then what does that imply about the computational complexity of the algorithm that I've thus far described?
Is it n to the 4?
Yeah, so that means that a naive algorithm for Viterbi, et cetera, maximum likelihood inference, has order of n to the 4th times m, the number of SNPs, the length of the HMM. And this is kind of bad, because, well, if n is 1,000, then n to the 4th is bigger than you would like. So that's where this clever thing about the transition matrix comes in. You actually don't need to pay n to the 4th. And there are, well, I'll tell you about 2 things that you can do to reduce that n to the 4th. So the first thing that you could observe is, well, where did this n to the 4th come from? It came from the fact that I have now n squared states in each column. And as I walk from one state to the next state, naively, I need to consider n squared possible transitions from each of the n squared states. That's where the n to the 4th comes from. Well, one very simple optimization that you could do is you could say, well, where did this n to the 4th come from? It's n squared times n squared, because I'm considering the possibility of switching both the paternal haplotype and the maternal haplotype to any other paternal haplotype and any other maternal haplotype at each position. I could just simplify my life and say I'm only ever allowed to switch one of them at a time. And that would be almost the same model and would get almost identical results. And that would actually reduce your cost immediately down to n cubed times m, because you have n squared states, but then each of them only kind of branches out to n other states. It turns out that you can actually do even better. You can actually go down to can go down to just order of n squared times m. And that you can do by exploiting the structure of that transition matrix. Basically, the fact that you're kind of, once you conditioned on your choice to jump, you're equally likely to jump to any other place. And the way in which, the easiest way, I think, to describe this idea is to imagine that we build ourselves a slightly fancier looking HMM that looks almost like the HMM that I just wrote down. But now I'm going to put something in between each column, each pair of columns. And I'm going to just call this state that I've added exactly one state in between each pair of consecutive columns. And this is called a jump state. And that just means I'm allowed to, or actually, sorry, what do I need to do? I need to, all of my states, pseudo states plus a jump state. And so now what does the new HMM look like? Well, I'm only allowed to transition down to this jump state, after which I can transition back to any of the actual states. And so what does that encode? It basically encodes the structure of this transition probability matrix that once you've gone down to a jump, then you can kind of, then it sort of doesn't matter where you came from. And then you can go right back you're equally likely to then choose any state for the next one. And well, what does this mean? It means that the number of arrows from any position to the next position is now only n squared, the number of states, or 2n squared, as opposed to n squared times n squared. And that takes down your running time to order of n squared times m again. So I believe I'm now out of time. Well, if anyone wants to hear more about HMMs or have any specific questions about phasing or imputation, feel free to come find me at the end of the session.
Thanks, John, for that introduction, for the invitation to speak to you guys, and also for introducing me to this community. It's been a really fun transition, hard sometimes, but also really gratifying. So the product I want to tell you about today concerns phasing and imputing repeat variants across the genome. And the context for this work is the problem of missing heritability, which presents a fundamental challenge in this era of GWAS. So heritability of a phenotype, which I'll denote by h squared, is the proportion of phenotypic variants attributable to additive genetic effects. Typically, this quantity is estimated from family or twin studies, that is by studying the phenotypic patterns in pedigrees where individuals have known relationships, known genetic relationships. On the other hand, there are genome-wide association studies which seek to identify the specific variants in the genome responsible for the inheritance of phenotypes. And theoretically, collectively, the variants found by the GWAS should explain the heritability observed in the pedigrees and the family studies. So if the heritability was properly estimated and the GWAS is large enough, the relevant variants should identify all of the heritability, and these two numbers should match up. And the conundrum is that across a broad array of phenotypes, h squared is just greater than h squared GWAS. That is, the heritability observed in family studies is quite a bit larger than the heritability explained by variants discovered in GWAS. For instance, recent GWAS of coronary artery disease explained about 21% of the expected heritability. A similar study of atrial fibrillation explained 11%, and even worse for schizophrenia, it was only 3.5% of the heritability. There are a lot of potential sources often cited for missing heritability. One possibility is that there are common causal variants of low effect size. And due to their low effect size, the GWAS simply have been underpowered to detect these effects. Another possibility is that the causal variants are actually rare, And again, this creates a problem of power if the GWAS is too small. A third possibility is that the causal variants weren't genotyped to begin with. This might include SNPs that aren't on the study microarray, or structural variants affecting large portions of the genome which are notoriously difficult to genotype. Another possibility is that the heritability was overestimated to begin with, and the list goes on. So in this talk, I'm going to tell you about some ongoing work which seeks to address the challenge of genotyping a certain class of structural variants with a view towards strengthening downstream association studies. Specifically, our team is investigating genomic repeats with a view towards explaining some portion of this elusive missing heritability. I'm going to address a class of repeat variants called variable number tandem repeats. So these are regions in the genome where a specific nucleotide sequence is repeated a number of times, which varies among individuals. So here's a picture of a VNTR. This is from Erdal et al.'s Nature Genetics 1999 paper showing a VNTR on the gene LPA, which sits on chromosome 6. This VNTR is going to be important for us. It'll be a recurring and motivating example, so we're going to return to it. Can you see my cursor? OK. Is there a pointer somewhere? Laser pointer?
I'll look, but
OK. So in this figure, you have two alleles of this gene.
well, I can't tell if this is what we want.
OK, never mind. It's not important. Thanks, John. So in this figure, you see two different alleles of LPA highlighted by a fluorescence imaging technique. The first allele, the top one, has 4 copies of the red-yellow KIV2 region repeated for spanning a total of 22 kilobases. The second allele has 20 copies of the same repeat spanning 110 kilobases. So this is exactly what we're talking about. The number of repeats is changing depending on the allele. So for this VNTR, the number of copies of this repeat is roughly normally distributed in the population, and that's what you should have in the back of your mind when you think about these VNTRs, and that's what maybe distinguishes it from other types of copy number variants. So there are several reasons that VNTRs represent an attractive target for further studies. So first of all, there's a lot of them. There are thousands of VNTRs spread across the genome. A 2015 survey of structural variants by Sudamont et al. In Nature found about 3,400 of these variants, and we recently estimated that VNTRs span about 5% of the human reference. A second point is that examples have already been implicated in several human diseases. So there's the VNTR on LPA that I just showed you is linked to cardiovascular diseases. A VNTR in the gene coding for a dopamine receptor on chromosome 11 has been linked to attention deficit disorder, and shorter repeats have been linked to Huntington's disease and Fragile X mental retardation. Finally, as a class, VNTRs remain largely unexplored, and so their true effect on disease heritability maybe isn't known yet. And in large part, this is due to difficulties genotyping. Another point is that these regions are thought to mutate fast, which can add some complications in modeling these regions in the genome. So as I hope to demonstrate today, certain advances, methodological advances in the proliferation of whole genome sequence data present a real opportunity to get a handle on these regions in the genome at a scale we haven't been able to do before. So summing up, the overarching goal for this project is to perform a comprehensive study of VNTRs across the genome and to assess the role that these regions and their variation play in heritable human diseases. And as I said, the main difficulty is that these regions are hard to genotype. Okay, so before presenting our methods, I want to spend a few minutes discussing our motivating example, LPA lipoprotein A. So lipoprotein A is a key player in the regulation of cholesterol. So here's a cartoon of LPA. LPA has two main parts. There's the apolipoprotein B part, which is depicted here in red, which is hydrophobic and tends to bind to cholesterol. And ApoB is covalently bound to another part of the protein called apolipoprotein A, which is hydrophilic. Each copy of this protein has a repeated KIV2 region sitting on the apolipoprotein A portion of the genome. So this is a ring of 114 amino acids repeated some number of times, and the number of times varies between individuals. So this is the KIV2 repeat on this cartoon of LPA. And Kringle is just a fancy word for ring or circle. So this variable size domain, the size of it is determined by a repeat in the gene encoding for the protein. So the length of a VNTR on LPA determines this Kringle size. Here's a cartoon of the gene LPA, which, as I said, sits on chromosome 6. There's the maternal and the paternal haplotypes. And each copy of this gene has a repeated KIV2 region consisting of a particular 5 kilobase sequence repeated somewhere between 10 and 50 times. In this cartoon, the maternal copy has C equals 4 copies of the repeat, and the paternal haplotype has C primed equals 6 copies of that same repeat. And then when this gene is transcribed, that corresponds to the number of red-yellow repeats you see in the figure below. One reason this particular VNTR is so interesting is that this copy number has been shown to be correlated with both protein concentration and heart disease. So the concentration of lipoprotein A, which I'll denote by LPA, is highly heritable. The family studies estimate that its heritability is between 70 and 90%, and inversely correlated with the LPA VNTR copy number. So here's just a sampling of the literature. So here's a plot of the LPA VNTR copy number against LPA concentration. And this has been documented kind of extensively in the literature. Whenever the copy number goes up, the protein concentration goes down. In addition, the copy numbers have been linked to cardiovascular disease. So here's a plot showing an increased risk of heart disease as for people with small VNTR copy numbers. Okay. And this is just a small sampling of the literature. Many people are studying this particular gene and this particular VNTR. This is the motivating example for our study. We're seeking to catalog VNTRs like LPA that are associated with heritable phenotypes. Maybe we're not going to find something quite this good, but maybe we'll find something good nonetheless. Okay. So as I said, a big challenge in studying these regions is the difficulty in genotyping. So now I'm going to review the literature on genotyping VNTRs. And first, there are the experimental techniques. These are generally low throughput and have been used to genotype VNTRs in small cohorts. So experimental protocols have been developed using gel electrophoresis, quantitative PCR, fluorescence imaging. Here's a picture from Kraft et al.'s Human Genetics 1992 paper where they used a gel assay to genotype VNTRs in 156 individuals. And here's the figure I showed you earlier of LPA using fluorescence imaging. Experiments like these yield VNTR genotypes in small cohorts, typically under 1,000, and were used in the 90s to establish the link between LPA, cholesterol levels, and heart disease. State-of-the-art methods estimate VNTR numbers, genotype VNTRs, from data in whole genome sequencing. So the reference here is Hansaker et al.'s 2015 Nature Genetics paper, where these authors gave a method to estimate diploid copy numbers, that is the sum C plus C primed across the two alleles, the VNTRs, from read depth in whole genome sequence data. So here's a figure depicting the author's method. The plot shows a normalized read depth and how it varies with genomic position near LPA. The normalized read depth increases in the KIV2 region and is variable between individuals. And the specific depth can be used to give an estimate of what C plus C primed is. So these methods were shown to be both accurate and precise, at least at LPA, in qPCR benchmarks. I should mention that Bob Hansaker is a key collaborator on our project and supplied us with all the diploid copy number estimates used in the analyses I'm going to tell you about. A second reference here is Zekavad et al.'s 2018 Nature Communications paper. These authors applied the methods in Hansaker et al.'s paper to genotype the LPA VNTR in a whole genome sequence cohort of 12,000 individuals. Using their genotyping, they built an imputation model for the LPA VNTR. The imputation model uses SNP genotypes near LPA to infer the diploid copy number. Here's a plot from their paper. The x-coordinate corresponds to the genotyped diploid copy number, C plus C prime, from the whole genome sequence data, and the y-coordinate represents what their model predicts for the same quantity. And their model succeeded in explaining about 61% of the variation in these diploid copy numbers. So while these papers represent important advances, there are a couple limitations. So one is that these methods supply no haplotype phase information. They only report on the sum C plus C primed of the two copy numbers without saying anything about C and C primed individually. A second limitation is that they require whole genome sequence data, which is expensive or of limited availability, or they require imputation and suffer inaccuracy as a result. So returning to our project goal, which I'll remind you was to study VNTRs across the genome and assess their role in disease. To build on these works of Zekavad et al. And Hansaker et al., what we're going to do is we're going to seek to model the haplotype phase structure in VNTRs, incorporate this additional structure to improve imputation accuracy. And I'll just remind you that a haplotype imputation typically outperforms diplotype imputation because haplotype matches in a given cohort are much more likely than diplotype matches. In addition to improving accuracy, the haplotype phase information itself is interesting for downstream analyses of non-additive and allelic effects of these regions. Okay, so here's my favorite mantra from my time at UChicago. So I'm about to tell you the theory how we phased and imputed these regions, but maybe this is a good time to pause if there are any questions. Yeah. So that's all well and good in practice, but how does it work in theory? How are we going to phase and impute these VNTRs? So our motivating problem is to incorporate the haplotype phase at VNTRs to estimate copy numbers. And we can rephrase this into two more concrete problems, a phasing problem and an imputation problem. So the phasing problem is to estimate the haploid copy numbers C and C primed in individuals with known diploid copy numbers C plus C primed and SNP genotypes. This type of data is now available in whole genome sequence cohorts thanks to the methods of Hansaker et al. The imputation problem, on the other hand, is to determine C and C primed using the SNP genotype data only. These two problems and their solutions are closely related. I'm going to primarily focus on phasing. I'll say a word about imputation at the end. But phasing really guides the imputation. So we can start by writing down the system of equations imposed by the diploid copy numbers. And when we do that, maybe we're a little daunted. The task appears challenging. So suppose we have a cohort of n individuals, each with a pair of copy numbers, Ci and Ci primed. Ci and Ci prime sum to a diploid measurement, di. And we know the di's, and we want to know Ci and Ci prime. This system of n equations has two unknowns, so it's underdetermined, and we can't really solve it with the information at hand. In particular, there's a consistent solution to these equations where any individual Ci, any individual copy number, is an arbitrary number and has nothing to do with individual i's genome. So what we're going to do is we're going to use structure in the population to turn the underdetermined system into a determined one. Specifically, we're going to look for haplotype sharing between individuals to impose additional constraints on this underdetermined system. So the key idea is to use the SNP genotypes near LPA or another VNTR to identify pairs of haplotypes which are likely to be identical by descent, IBD, at the VNTR. We can then infer likely equalities between copy numbers, additional equalities between copy numbers in the individuals. For instance, if we estimate that individual 5's first haplotype is identical by descent with individual 3's second haplotype, we might conclude that C5 is C3 primed. So in broad strokes, that's all we're doing. The challenge is to probabilistically and carefully weight the additional constraints to come up with the best possible estimate for Ci and Ci prime. I'll just say, so with the extra constraints, we expect and hope that that underdetermined system becomes determined. For instance, if there's 1,000 individuals with 2,000 haplotypes, but only 100 VNTR alleles floating around, then the 1,000 diploid measurements are likely to determine those 100 numbers. Any questions about that? Okay. So our methods for estimating haplotype sharing are based on the Lee-Stevens framework, which Paru just told us about. So I'm going to spend a few minutes kind of recapping that method and telling you about it very briefly. So the heart of the Lee-Stevens approach is a generative model for constructing new genomes from a reference panel of known phased haplotypes. So here's a panel of known haplotypes. Lee and Stevens define a probability distribution over new haplotypes given such a panel. So haplotypes are represented as sequences of zeros and ones corresponding to the reference and alternate allele at each SNP. A typical new haplotype is a mosaic built out of long stretches of reference haplotypes with a small number of jumps between panel entries. The jumps are meant to model recombination events, and the model preference for contiguous stretches encodes the local correlations and linkage disequilibrium we often see in genotypes. The precise model is specified by a hidden Markov model, where the hidden variable corresponds to the shared haplotype at a given location in the genome. A new diploid genome is sampled simply by drawing two haplotypes independently. So given a panel of known SNP haplotypes and a new individual with diploid information only, that is a sequence of zeros, ones, and twos, we can condition the model on the known information. So the result is a probability distribution over haplotypes given the panel and the constraints imposed by the diploid information. This conditional distribution then dictates an expected phase for the new diploid individual, and this expected phase can be computed efficiently using hidden Markov model techniques. So summarizing, given a panel of known haplotypes and an individual with diploid information only, the Lee-Stevens generative model provides a recipe for estimating the phase of that new individual. To phase an entire unphased panel of diploid individuals, we start with a random initial phase, then cycle through the individuals, re-estimating the phase of each individual using the remainder of the panel as reference. So here's the panel with an initial random phase. We start with individual I, and we forget their phasing. We then use the remainder of the panel to estimate individual I's phase and update, and then cycle through the individuals. And then we just cycle through the individuals until the answers converge. Okay, so that in a nutshell, that's the Lee-Stevens approach to SNP phasing. That's how you take a collection of diploid individuals and come up with an estimate for their phase. Now we want to go from SNPs to copy numbers. So to phase copy numbers, we're going to adapt this Lee-Stevens generative model to a model for the VNTR setting. So for SNP phasing, the haplotypes were represented by sequences of zeros and ones. In the VNTR setting, each haplotype consists of a SNP sequence of zeros and ones surrounding the VNTR, together with a VNTR copy number. So given a panel of VNTR haplotypes, we need to describe a method to generate new haplotypes and diplotypes. So to generate SNPs, our model is literally identical to the original Lee and Stevens model. The VNTR copy number is then drawn from a normal distribution centered at the known copy number of the shared haplotype dictated by the hidden variable in the Lee-Stevens HMM at the VNTR. The result is a probability distribution over SNPs and the VNTR copy number. So I thought here's a moment where I might write something down. So we're trying to, we want to give a probability distribution on copy numbers and haplotypes given a panel. And that's going to be this sum over the haplotypes in the panel of the probability of C and H, and that the hidden variable is equal to the haplotype little h given the panel. Okay, so this is the haplotype is shared with haplotype H. At the VNTR. Okay. And this is just the sum over the haplotypes of the normal Lee-Stevens probability that can be computed by HMM techniques, and then the probability of choosing C given that the shared haplotype was H. And what I just said is that C should be drawn from a normal distribution whose center is the copy number of haplotype H with some variance, which is a parameter in the model. Okay. So that's a way of taking a panel consisting of SNP genotypes and copy numbers and spitting out new VNTR haplotypes. Any questions about that? And then a diplotype is just two independently sampled haplotypes. And once this probability distribution is set, the math really dictates the rest. To spell this out a bit more, given a new individual with known SNP haplotypes and diploid copy number, the model dictates posterior distribution on copy numbers. And then taking expectations yields a phase estimate for this data.
Ronen?
Yeah.
The noise on the normal distribution here,
yes.
Is that learned, or is that something you fix?
That is tuned, and I'll explain how we tune it in a bit. If I haven't answered your question by the end of the talk, please ask it again. And for imputation, I said phasing guides imputation. Imputation, we do the same thing. We're just conditioning on less information because we don't have the diploid copy number anymore, and no iteration is required. Okay. So that, in a nutshell, is our approach to phasing and imputing. After working with data, we found that the Lee-Stevens inspired approach performed well at the LPA VNTR, but it seemed to overweight the best matches, which resulted in a slightly reduced performance. I'll tell you exactly how we benchmarked our method in a moment. So we found that performance improved considerably after replacing the Lee-Stevens HMM probabilities with simpler scores based on identical by state length, just the amount of the SNP genotype the pairs of haplotypes share around the VNTR. This remains a bit of a mystery. It seems that sometimes the good but not the best Lee-Stevens matches are the real matches at these VNTRs. The IBS approach had other advantages in runtime, which allowed for easier tuning of parameters. So in the end, we settled on this approach. Okay. Enough theory. How did we benchmark our methods, and how well did we do? So to benchmark our results, we needed to generate a collection of gold standard data. And to do so, we used whole genome sequence data in quartets. Specifically, we used data from the Simon Simplex collection, which is a study of autism involving 519 quartet families. Each family consists of 2 parents and 2 children, and each participant was whole genome sequenced. Our use of this data was approved by the Simons Foundation for Autism Research for an ongoing study of the role that structural variants play in psychiatric disorders. So the reason this is so useful is that for the IBD1 quartets, that's the quartets where the siblings share exactly 1 copy of the VNTR, we can solve exactly for all their copy numbers. So here's a quartet. We've got 4 individuals with 4 diploid copy number estimates. And it looks like we have 8 haplotypes, but we really only have 4 haplotypes because there's haplotypes sharing between the parents and the kids. So we have a system of 4 equations and 4 unknowns, and it just so happens that if the kids share exactly 1 copy of the VNTR, we can solve this system of equations exactly. We expect about 50% of the quartets behave like this. There were 266 at LPA, resulting in 1,064 haploid copy numbers. So our phasing benchmark is to phase the parents and then compare the results with the gold standard data computed in these quartets. Any questions about that? Okay. And to John's question, we used the remaining quartets, the children in the remaining quartets, to tune the model parameters, like that mutation rate. There are a couple other parameters. So we chose parameters which minimize the squared error of the additional relations imposed by the children in these other quartets. And this technique was helpful because it avoided overfitting towards our IBD1 benchmark. So hopefully our IBD1 benchmark is actually pretty accurate. Yeah.
Would you say that these 4 equations and 4 unknowns is where your algebraic geometry background was most essential?
Absolutely. Yeah. Okay. Right, avoids overfitting. Okay, so how did we do? Well, so we performed that benchmark. We phased the parents, and we compared to the IBD1 gold standard. And this is what we got at LPA. So our benchmark indicates that our phasing algorithm predicts 87% of the variation of haploid copy numbers at the LPA VNTR. So here I've plotted the copy number inferred from the IBD1 quartet versus our phasing model's estimate of that same copy number. We also estimated our imputation accuracy. So this is a slightly different experiment. In a slightly different sample. Here we use data from the genomic psychiatry cohort, which is a whole genome sequence cohort of 3,500 individuals. We performed an experiment similar to what Zekavad et al. Did in their paper. So we phased the LPA VNTR on 90% of this cohort using the diploid copy number, and then used the results to impute the diploid copy number on the remaining 10% of the cohort without using their diploid copy numbers. We then compared the imputed predictions for the diploid copy number with a true diploid copy number as estimated from the whole genome sequence data. Okay. So the estimate for diploid copy number is on the x-axis from whole genome sequence data, and our model's best guess is on the y-axis. And our imputation models seem to explain about 80% of the variance here. So this, you can sort of compare to the 61% in Zekavad et al.'s diploid imputation, with the caveat that these are really different experiments. We worked with different populations, different sizes, and different ancestries. So you can't, similar but different. Yeah?
Unexplained variance can come from a lot of places,
Mm-hmm.
obviously including like measurement error and the diploid copy numbers and imputation error and de novo mutations as well. Do you guys know what the de novo mutation rate is for, I guess, I don't know what you'd call it, for extending or decreasing the length of the repeat,
Yeah.
and like how much can we do to that?
I don't have a specific number for you. These regions are thought to mutate faster than other regions in the genome. So perhaps the number is quite high. The fact that we're phasing pretty well indicates to me that it's not all that high. Because if it were really very, very high, we wouldn't find haplotype matches. And the fact that we can phase at 90% indicates, at least at this VNTR, it's not all that fast. Yeah. Other questions? Okay, so we're about to expand our horizons and go beyond LPA and turn to VNTRs across the genome. So I'll remind you there are about 3,400 VNTRs spread across the genome. We recently genotyped and phased VNTR copy numbers for about 3,100 of these VNTRs. And we ran the same SSC IBD1 benchmark that I just described for LPA. So I'm not going to tell you the results of this experiment. Before I show the plot, let me tell you an important point, which is that the diploid, so far I've kind of been implicitly assuming that the diploid VNTR genotyping from whole genome sequence data was accurate. And that's definitely true at LPA, but it's not as true at some other VNTRs, and it kind of varies considerably depending on the specific locus. One way to get a sense of that is to look at the correlation in copy numbers in the IBD2 siblings. That sort of gives a measure of how accurate the genotyping is. So here's the sibling correlation at LPA. So here, these are just the IBD2 quartets, the quartets where the children have the exact same 2 copies of LPA. And we've plotted sibling 1's diploid copy number estimated from whole genome sequence data and against sibling 2's copy number. And you see these are very strongly correlated. The R-squared is 0.95. So this continues to be true at some VNTRs and just isn't true at other VNTRs. Here's a VNTR on chromosome 1, where the diploid estimation just isn't nearly as precise. The sibling 1's correlation with sibling 2 has an R-squared of 10%. And as you might expect, we're probably not going to be able to do as well at VNTRs like that. Also, at VNTRs like this, the benchmark might be highly skewed because the way we were generating this gold standard data was from these diploid copy numbers. So if the diploid copy numbers are unreliable, the gold standard might not be very reliable either. Okay, so here are VNTRs genome-wide. So on the bottom, I've plotted this IBD2 sibling R-squared that I just showed you. LPA was very high, and that VNTR in chromosome 1 was very low. And against that, I've plotted the result of our phasing benchmark, R-squared. And this red dot up here is the LPA VNTR, where we had a high IBD2 sibling correlation and a high phasing benchmark. Okay. So just to get our bearings, the things at the top right we're very happy with. The things on the bottom left, the whole genome sequence diploid measurements didn't do so well, and so maybe there's not much you can do with phasing. And the things on the bottom right, well, phasing, something's going awry with phasing. Summarizing that a bit, for about 1,000 of the VNTRs, we managed to phase with an accuracy of at least an R-squared of 0.6. And then with an R-squared of 0.8, we got 371 VNTRs and about 100 for phased with an R-squared of greater than 0.9. Okay. We've begun to identify several regions in this bottom right corner why our phasing method is failing. In the interest of time, I'll be very brief here. There are some VNTRs that behave like rare SNPs. So this is a VNTR. This is the IBD2 sibling correlation plot. And you can see most of the variance in this VNTR is due to this one data point, this one rare allele up at the top right corner. And if the VNTR is behaving more like a rare SNP, maybe we're going to have more trouble phasing it. Maybe even that rare allele doesn't even occur in the IBD1 quartets. We also had kind of mixed success at long VNTRs. So here's the same plot, just kind of squished, that I showed earlier of IBD2 sibling R-squared versus phasing R-squared. And I've highlighted in red the VNTRs we expect to do well on, that there's a high IBD2 sibling correlation, but the VNTRs are also long in the human reference genome. And you can kind of see they're all over the map, and they account for a lot of the VNTRs in our bottom right corner. Here's that same high IBD2 greater than 0.8 bit of the plot, but here I've plotted the log VNTR length versus the phasing accuracy, and you can see there's kind of this, it does well for a while, and then once the VNTR is long enough, results are kind of all over the place. Okay, so a different approach might be required for these loci. Okay. Now our team is moving beyond SSC parents, which was a cohort of 1,000 individuals. And we're imputing copy numbers into the UK Biobank, a sample much larger, 500,000 individuals. So this is an exciting time for the project. This data set provides a rich target for imputation for several reasons. It's large. There are 500,000 participants, predominantly white and British. Each participant was genotyped with a DNA microarray, and there's extensive phenotypic information available for these individuals, including electronic medical records and blood lipid levels. So we've now begun to impute VNTR copy numbers into the UK Biobank. So far, we've imputed the LPA VNTR and 15 other VNTRs, which phasing was doing pretty well on. And so this is all kind of preliminary. I don't have much to tell you on this yet. We can revisit, because we have blood lipid levels and LPA VNTR copy numbers, we can revisit this correlation between LPA VNTR copy number and lipoprotein A. So here I've plotted the log of the lipoprotein A concentration against our imputed diploid copy number using the diploid, the imputation model I've been describing. And we found that the imputed diploid copy number accounts for 18.5% of the variance in log LPA. And at least, so something's going right here. I'll show you one more plot. One advantage of our imputation model over other models is that, as I pointed out earlier, our model gives haplotype information. So we can investigate how each allele separately affects lipoprotein A. So here I've plotted log lipoprotein A concentration versus 1 over the minimum across the two alleles of the copy numbers, again, imputed from our model. And there's a significant bump in the amount of variance explained. It went from 18.5% to 25.7%. This is probably not news to the experts in LPA, but it's a reflection of the complicated genetic architecture of LPA, and we're excited to use our results and this new data to help us understand this locus. Okay, any questions about that?
Is it, I mean, biologically, are you taking the min because you think that the driver here is the protein with fewer loops?
Yeah. And I think that's an observation that's been made, in the literature in the past. And I think there are often cases where only one of the alleles is actually expressed, and it's often the minimum allele. Yeah.
So if you didn't do the imputation and you use SNPs to explain the LPA, what R square would you have got?
I don't know about using all SNPs, but there are definitely SNPs that tag LPA very strongly. There's a SNP that has an R squared, what was it, 0.4? Do you remember? 0.37? 0.28 on the log scale. Which we suspect is a mixture of that SNP tagging the VNTR copy number and maybe tagging some other causal variant. But there's definitely, some other data points I can give you. In GWAS of this trait, there are lots of significant SNPs and even lots of significant SNPs after conditioning on the LPA copy number. And one thing we're excited to explore is how all of these things interact. Okay. Since I'm running out of time, I'll just wrap things up by telling you some of our plans for the future. The first thing we're going to do is we're going to scale up. We're going to impute VNTRs across the genome in the UK Biobank. And we're going to perform a host of VNTR association tests with that broad array of phenotypes that are available in the UK Biobank. We're also going to try to estimate the total contribution of VNTRs to disease heritability. And we'll explore lipoprotein A more. We'll seek to refine the genetic architecture of this very interesting trait. Okay, so to remind you what I've told you, so using a Lee-Stevens-inspired approach, we can phase VNTR copy numbers in whole genome sequence cohorts and impute VNTR copy numbers into SNP array data. In SSC parents, we managed to phase about 1,000 VNTRs with an R squared of greater than 0.6, and we phase the LPA VNTR of specific interest with an R squared of 0.87. We can impute the LPA VNTR into GPC with an R squared of 0.8, and our imputed LPA VNTR copy numbers in the UK Biobank recapitulate the well-known link between the VNTR on LPA and lipoprotein A concentration. And with that, I'd just like to acknowledge my collaborators, Parulo, who's been working with me very closely on this project, and Bob Hansaker, who's been instrumental in using these whole genome sequence methods for genotyping diploid copy numbers. Thanks for listening.
Thanks, Ronen. So I'll ask one brief question,
Yeah.
then we'll break, come to discussion. When do you think you might have a preprint on some of this?
This year?
Good answer. Well, we really appreciate you sharing it even before that. That's kind of the ethos we want to promote. All right, so we'll have some more discussion in the front in a couple minutes. Anyone who wants to get on with their day, have a good day.